Hello everyone. Today we are going to discuss about the uh, how does an enzyme actually works and on what energy it depends to work to perform the catalysis of a substrate to convert it into product, and how does the substrate itself also work on itself to get participate in a reaction to form a product. So let's move toward these things and since enzyme is actually involved in each and every reaction of our body, from digestion to absorption to muscles contraction via actin and myosin filament in muscles to everything. Actual enzyme is present everywhere and it's actually involved in each and every reaction that is taking place within the body of a living being. So suppose this is an enzyme which is have a bold shape of morphology, and here is the active site. This is the substrate for this bold shape enzyme. And now, in my previous lecture, I have discussed about the enzyme substrate complex, the enzyme substrate complex form, and then it gives a product. But prior to the formation of an enzyme and substrate complex, prior to the commencement of a reaction that is catalyzed by an enzyme, a substrate itself have to uh, get modified mechanically, structurally, or uh, the intra-chemical bond within the substrate also get modified so that it can it can bind to the active site of the enzyme accordingly. So this substrate, suppose this is the actual region of the substrate with which have to get combined with the enzyme to form its product. Now let's move toward the what actually happens with the substrate. So as we know that each and every uh, each and every living uh, biomolecule within the living system is having energy. Either it is less or higher, either it is a substrate or a product. Each and every molecule have some energy, which is usually denoted by G. But to discriminate between the products and the substrate energy, I use this G dash for the product's energy. Now, for this to remain, for substrate and product to remain in equilibrium, the difference of these energy is required, and the result that obtained after this difference should be negative. More negative value, more the system is balanced, or more the reaction will uh, takes place feasibly for the product formation. So it's pretty obvious. So it can be happen only when the product's energy is lesser than that of the substrate energy. So this is about the energy. So suppose here we have a conical flask in which a reaction is taking place with certain molecule. This is the molecule that is present within the uh, flask. And now before participating in any reaction with the enzyme to form product, this structure of the substrate also get modified either via breakage of certain bonds, formation of new bond within it, or electric charge transfer. Electrostatically, it get modified to raise itself up to a certain higher energy level. And this higher energy level is called transition state. This higher energy level is called transition state, as the substrate for the reaction are present at the ground state. For the forward reaction, this is the substrate S, which is present at some ground state, and to achieve the condition or the transition state, which is present at this point, top point of this hill, to achieve it, the substrate have to uh, raise up to this energy level. And if we talk about the backward reaction, then this product have to achieve this much energy to raise at itself at this point, so that it can easily participate easily participate in the reaction. And this the energy difference of these transition state is actually the activation energy. Till now, there is no enzyme involved. Only the substrate itself modifies itself. Also, one more thing that I missed at this point. This is the reaction. X-axis is the reaction, and Y-axis is actually representing the energy. Sorry, I'm not. It is not being written in this slide, but that's why I'm explaining it verbally. The x-axis is actually the reaction, and this y-axis is energy. So the substrate or product in backward reaction, they have to modify itself to raise up to this energy level, so that it can easily get involved in with the enzyme to form final product. And the difference of this energy, the transition state, is called as activation energy, that an enzyme have to lower down to form a product. Please don't get confused with the enzyme substrate complex and this transition state, because there is the enzyme substrate nothing to do with this transition state. Transition state is only and only for the substrate, that it achieve after some chemical structural modification within its structure. So now let's move toward, after achieving this transition state, what will happen. This is the enzyme, and since we know that enzyme is made up of protein, so this is the internal structure with different amino acids in a chain, and with different R group. Now a substrate after modification start to combine with the enzyme either via some covalent bond that is formed between the catalytic group and the substrate. Although they are transient, they are not permanent, because a substrate have to get converted into product, and an enzyme will leave the reaction in its native form. Along with this covalent bond, some non-covalent bonds also formation will non-covalent bond formation will also takes place between the substrate and the enzyme, and the energy that is released after this bond formations, covalent and non-covalent bond formation, this energy is called as, and the combination of the energy is called as binding energy. This binding energy is actually an enzyme used for lowering this activation energy for a particular reaction. So actually binding energy, we can say, is the source for enzyme to lower the activation energy. Now, so this was all about the substrate modification, enzyme, how does an enzyme work for a reaction, lowers the activation energy for any particular chemical reaction. So this is all about this, how does an enzyme actually work, and how does a reaction get started with a enzyme, and how the substrate participate or modified itself before participating in a reaction. In the last, here are some questions related to my previous lecture on enzymology, and to this part of this enzymology. Please solve them, and if you found any issue by solving them, please mention it in the comment box. And if you like this lecture, please mention in the comment box, and hit the like button, subscribe my channel, share it as possible as you can. And if you find some loopholes in the lecture, please mention that too, so that I can modify accordingly, and it will also motivates me. Thank you.